Triacylglycerides or triacylglycerols are a long-term energy storage in molecules. So their catabolism involves hydrolysis to glycerol and fatty acids, and then subsequent beta-oxidation of fatty acids. The triacylglycerides are carried by lipoproteins to tissues where they are hydrolyzed to glycerols and fatty acids. Beta-oxidation of fatty acids occurs in mitochondria and, to a lesser extent and following a slightly different pathway, in peroxisomes. First step in catabolism of triacylglycerides is their hydrolysis. So, hydrolysis occurs extracellularly and is catalyzed by enzyme lipoprotein lipase, which is attached to the outer surface of the cell. So it's extracellular process. Triacylglyceride is converted to glycerol and 3 molecules of fatty acid. Free fatty acids are actually toxic as they are detergents and would disrupt cell membrane if they were present in free form in significant amount. So upon release, they immediately enter the cell where they are either metabolized or re-esterified and stored as triacylglycerides. They are also fatty acids are the main source of energy for the heart muscle, which utilizes fatty acids in preference to carbohydrates. So upon being released from triacylglyceride, so after hydrolysis and after entering the cell, fatty acid is activated because it has to be activated before it can be oxidatively degraded. The degradation is beta oxidation, as you'll see in a minute. So first step is a reaction catalyzed by acyl coenzyme A synthase, and that reaction requires ATP. So fatty acid is converted to the corresponding acyl coenzyme A. Acyl coenzyme A synthases that are specific for longer chain fatty acids function in cooperation with membrane transport proteins, so that fatty acid is activated as it enters the cell. Once it's bound to coenzyme A, it cannot leave the cell and is committed to metabolism. So esterification to coenzyme A commits fatty acids to metabolism. Fatty acids are activated in the cytosol, but the oxidation itself takes place in mitochondria. So carnitine shuttle transports acyl coenzyme A from the cytosol into mitochondria, where beta oxidation takes place. So beta oxidation is a metabolic pathway for degradation of fatty acids. Fatty acid, as already mentioned, enters the cycle as acyl coenzyme A, and products of beta oxidation are acetyl coenzyme A, which enters the citric acid cycle, and acyl coenzyme A, which is truncated by 2 carbon atoms. So it's a spiral pathway, and each cycle of the pathway shortens the fatty acid by 2 atoms. Each cycle releases one acetyl coenzyme A, which enters the citric acid cycle and leaves shorter and shorter chain. So, for example, it takes 8 cycles to completely degrade stearic acid, that's C18 fatty acid, and so there would be 9 2-carbon units, 9 acetyl coenzyme units, and of course final step produces two molecules of acetyl coenzyme A, so the last 2-carbon chain that is left also enters citric acid cycle. There is no need to carry out one more cycle. So the number of cycles that is needed to completely degrade a saturated fatty acid is number of carbon atoms divided by 2 minus 1. So in case of C18 fatty acid, that would be 18 divided by 2, 9 minus 1, 8. When we consider beta oxidation itself, it is shown here. It is a 4-reaction cycle and is essentially a reverse of fatty acid biosynthesis. In the first step, dehydrogenase, so acyl coenzyme A dehydrogenase, generates a trans alpha beta unsaturated coenzyme ester. A molecule FAD is the oxidizing agent, and it is actually catalytic oxidizing agent. It is regenerated by reaction with ubiquinone. So fatty acid oxidation provides electrons for oxidative phosphorylation because ubiquinone, as you may recall, donates then electrons in the next step to complex 3 of electron transport chain. So that's how fatty acid oxidation provides electrons to fatty acid to electron transport chain. Next, water is added across the double bond of the alpha-beta unsaturated acid, and reaction is catalyzed by enzyme hydratase. Third step corresponds to is actually the second oxidation step of the sequence. So third step overall is the second oxidation step, and it involves NAD as the oxidizing agent. So it's the cofactor. It's the stoichiometric cofactor. Reaction product is corresponding beta-keto coenzyme thioester, and reduced NAD, NADH is formed. It's released. In the final step is actually thiolysis, which requires molecule of coenzyme A, and it cleaves, it's basically a retroaldol reaction. It cleaves carbon-carbon bond to give molecule of acetyl coenzyme A, which is the one that enters the citric acid cycle, while remaining acyl coenzyme A is 2 carbons shorter compared to the starting acid and can undergo another 4-reaction cycle, and after each 4-reaction cycle it's shorter and shorter by 2 carbon atoms until at the end we are left with two carbon fragments, 2 acetyl coenzyme A fragments, which both enter the citric acid cycle. When we consider the overall process, each cycle of beta oxidation generates 1 molecule of dihydroubiquinone, 1 molecule of NADH, and 1 molecule of acetyl coenzyme A. Dihydroubiquinone, in the course of oxidative phosphorylation, generates 1.5 molecules of ATP. A molecule of NADH, in the course of oxidative phosphorylation, generates 2.5 molecules of ATP. And finally, acetyl coenzyme A enters the citric acid cycle. In the course of citric acid cycle, it generates 3 molecules of NADH, which correspond to 7.5 molecules of ATP after oxidative phosphorylation, 1 molecule of dihydroubiquinone, which means another 1.5 molecule of ATP, and 1 molecule of GTP, which corresponds to 1 molecule of ATP. So overall, in total, 14 molecules of ATP are generated in each cycle of beta oxidation. When we consider regulation of beta oxidation, it's primarily regulated by the availability of free coenzyme A, which is needed to make acyl coenzyme A, so activated acid for beta oxidation. And also it's regulated by the ratios of NAD to NADH and ubiquinone to dihydroubiquinone. And some of the enzymes of beta oxidation are also regulated by the product inhibition. Unsaturated fatty acids contain cis double bonds, and cis double bonds are obstacles to beta oxidation. An example is shown here, linoleic acid, and that shows how these obstacles are bypassed. So, of course, fatty acid has to be activated as coenzyme A ester, and then first 3 cycles of beta oxidation proceed as usual. Now, the 4th cycle cannot proceed as now there is a cis double bond in position 3, and so beta oxidation is not possible. Enzyme enoyl coenzyme A isomerizes double bond from position 3, so it isomerizes cis 3 double bond into trans 2 double bond, and trans 2 double bond is acyl coenzyme A, which actually is an intermediate of beta oxidation. If you look at beta oxidation on the previous slide, you can see this is an intermediate. And so Next step would be hydration, then oxidation, and then breaking of carbon-carbon double bond in retroaldol reaction. So now beta oxidation can continue. So fourth cycle of beta oxidation continues, and then we have 2 more carbons, and so fifth cycle can start with dehydrogenation. But once fifth cycle occurs with dehydrogenation, now we have 2,4-dienoyl coenzyme A. And so now double bond in position 4 is both cis and in position that would interfere with addition of water to double bond 2, because actually addition of water that is now supposed to occur to trans double bond would actually occur at the end of the system. It would occur on the double bond shown in red. And so for that reason, reduction has to happen. So NADPH is stoichiometric reagent, and enzyme reductase reduces 1,4 double bond system. So it reduces conjugated double bonds. It's 1,4 addition of 2 hydrogens. And upon reduction, we end up with trans double bond in position 3. That double bond is isomerized to position 2, and now oxidation can continue. And finally, the entire chain is oxidized. As a result of these bypass reactions, unsaturated fatty acid yields less energy compared to a saturated fatty acid. Each double bond that is present means that dehydrogenation reaction, which generates dihydroubiquinone and 1.5 molecules of ATP, is not necessary, so it doesn't happen. And also NADPH has to be utilized for reduction, and that consumes additional 2.5 molecules of ATP. So total of 4 molecules of ATP are consumed. So for each double bond, for each cycle that involves a double bond, instead of 14 molecules of ATP that are generated, only 10 molecules of ATP are generated, or you can view it that for each double bond, 4 fewer molecules of ATP are generated. So that completes our brief examination of catabolism of fatty acids.